So yeah, we welcome any comments on what people think, having heard these series of talks. A lot of discussions about using the mouse models and looking at potential therapeutics, moving from the mouse to the human with all of the limitations or advantages. We have some experts here who have presented some elegant work in various animal models, and I wondered what your thoughts are. Obviously, Ulrika has described some of the early preclinical work on this pathway. Tom has compared two limbs of the growth hormone IGF-1 pathway. What is the potential for these models to move into human development?
Well, I think I'd probably put my money behind myostatin and activin pathways for bone and muscle. I think, I mentioned limitations. I mean, if there are people who are using growth hormone, and obviously the worry with activating IGF-1 is the underlying, effects you'd have on, malignant cell formation. There's all kinds of problems that you might get into. Now, if again you could get to mechanisms that would be uniquely stimulated by growth hormone to enhance insulin sensitivity at the target tissue, if you could really dissect that pathway and separate it from anything else, then that would be with a small molecule, for example. But that's challenging. I think that's where that field lies, and I can't imagine a drug company, you can comment on that, going after that pathway as opposed to the what I think are more reasonable ones where you can actually affect through either soluble receptors or antibodies these ligands that are inhibitory, basically by inhibiting their action.
I think in general, if you look into the drug industry, 80 or 90% are inhibitors. It's much easier to control. But I think if you want to go with the IGF-1, you really have to go into situations where IGF-1 is low, because you cannot afford to go above physiological levels. And what we have learned from our animal models is, for aging, rat is a good model, mouse is a bad model. Mouse in the muscle, they just die, they don't get atrophy. They don't get sarcopenia. So you have to wait till 27 and 30 months, and it's really difficult. And what we have also learned is, this is why we put so much efforts in the human cellular system at least, and try to get correlative human tissue data, because there are differences. I've just had a target, it's working beautifully in human muscle cells, it's not working at all in mouse. Lessons learned.
Candice Dahimik, San Francisco Veterans Affairs. Dr. Kiel, I was unable to actually pose a comment and actually a question regarding your talk. Now, in your presentation, you actually provided information on genome-wide association studies for bone and muscle. Now, my question is that in the methodology, as you tried to make a list of these genes, did you take into consideration the effect of imprinting status of these particular genes? In your first, I think, trial involving 1,000 or so something samples, you didn't find any hits, and I was wondering whether the SNPs were the effect of the SNPs were basically masked by the imprinting status of the patient or the person involved. Meaning, for example, you could have an effect of a particular SNP if that locus is actually actively transcribed, as opposed to the SNP being in an imprinted region and it's shut down. So you don't have biallelic expression. You have monoallelic expression, for example, paternally imprinted or maternally imprinted genes. Could you comment on that?
Yeah. I think these studies in humans in large samples are only the first step in identifying variants that might affect actual expression. And so the next step is to combine that with some functional studies which are underway, looking at expression, QTLs, and actual functional implications of some of this. I don't think the small first genome-wide association studies suffered from this problem as much as just trying to find very small effects with very small sample sizes.
I see. Well, are you also are you familiar with any study that would actually analyze promoter regions, checking for methylation status and the correlation between aberrant osteogenic or perhaps myogenic development?
Yep, there's certainly studies underway to do whole genome methylation. Some of this is very tissue specific and is made maybe more progress in animals than in humans, and it raises the complexity of this is an order of magnitude greater than just studying genetic variants, I think you're correctly pointing out.
Okay, thank you.
Hey, thank you very much. My name is Sean Garvey. I'm from Abbott Nutrition, and in our research program, we study sarcopenia. We're looking at 24-month-old Sprague Dawley rats, and we were recently encouraged by a paper published by Merck, and they recently published, similarly, the Sprague Dawley model. And there's some really interesting data in that paper, and this is more specifically targeted to the scientists at Novartis. They showed that not all rats age similarly, and almost making them an even better model for human sarcopenia. And what's interesting is they associate high-functioning rats with this fiber type 1 clustering, the idea being that type 2 fibers atrophy, apoptose, and there might be some regeneration of type 1 fibers. And I'm curious if you can elaborate any more on the specific rat model you use, and if you've looked at type 1 and type 2 fiber population.
First of all, we have two sites. We have one site in Basel and one in Europe and one site in Cambridge, and we have two different strains, and we could reproduce the data. This is two different strains from two different providers. But we know that it's a little bit is a variability. And also how you get these animals, if they really have a date birth and if it's outbred or inbred, so it's very, very difficult. So what we are looking into is really have other readouts, like we are measuring mitochondrial activity, things like that. We start that now. But we know it's very, very difficult to go from a rat model to a human anyway, and we have a problem with variability as well. And we have done a full fiber type analysis, and they change, but I don't know each of the details, but we have done all that, and they change.
Beth Lee, Ohio State. In Dr. Kiel's talk, one of the genes that really popped out for me in your analysis that associated with both the bone and the muscle phenotype was GRB10. It works downstream of the IGF receptor and is thought to inhibit growth and then also interact with AKT signaling. And I was just wondering if any of you have done any dissection on that arena.
I saw that as well.
Thank you. This is part of the collaboration. Thank you for that comment. GRV10.
GRB10.
Yeah. GRB10.
Yeah, it's been. I mean, I've done a little bit with this in other tissues, but when knocked out, it actually promotes growth. The animals get much larger, and it does work down of IGF receptor. And as I said, in some cell types, including some of the ones I've worked in, I just published a paper on it. It interacts with AKT and aids in its activation. So this introduces some of the questions that we had for future research directions, things that you're suggesting now. How do we move from what we find without any hypotheses to you're already putting together a few pathway from your own information? Is this something that will likely be feasible going forward? I mean, we can go down the list and come up with some other ideas as well. Yeah, I mean, I have ideas about it. But yeah, I mean, there's null mice out there, and as I said, I've been looking at it in a different tissue completely in kidney. But it's an interesting protein because there's a lot of actually splice variants. Nobody knows what they do. This is a protein that's got a lot of room for study, and it's just not really well looked at this point.
The NIAMS has issued follow-up opportunities to take some of these initial discovery work and move it into exactly the kind of thing you're saying. So I think there's a lot of potential. One thing that we've done is we had one of the BERT grants that was mentioned during lunchtime. And so we took some of our top-ranked bone findings, and we did a screening, shRNA knockdown library screening of some of the novel ones that we didn't know had any effect in bone using human mesenchymal stem cells and osteoclasts to try to identify whether there's any functional significance knocking down these genes. And we're still making some headway there, but candidates like the one that you mentioned could be examined that way to start out.
And actually, as long as you're speaking about osteoclasts, we just finished a study too that I noticed one of your hits for the bone study was Myo9B, and we're just finishing up a study on that. We're about ready to submit. Myo9B is a myosin that contains a row gap domain and seems to be the major row gap for regulating cytoskeletal organization in osteoclasts. So I just thought I'd bring that up.
And could you again say your name? Did I miss that?
Beth Lee from Ohio State. I'd like to talk to you later, actually.
Ohio State.
The Ohio State, yes.
Thank you.
Thank you.
But this is exactly how you learn the most, if you compare different studies. This is what we did. We did our in vitro studies in vivo and then compared and looked into overlaps and how they relate to each other. I think this is how you most about the pathways.
Did you have a question, Steve?
Dr. Cummings, did you have a question? No?
Okay.
You know, I just wanted to measure another.
Just a short question.
Oh.
Okay. And ubiquitin ligase plays an important role in muscle atrophy. How about ubiquitin ligase in bone disease? Dr. David?
So the ubiquitin ligases have a long history in muscle atrophy. So I guess one of the questions, and I'm going to throw this open to the bone people, is are there bone-specific ligases and/or are there ligases directly linked to osteoporosis and that kind of thing?
SMERF1 was one of them. This is a smart ligase, a hacked ligase, and I think it has a bone phenotype. SMERF, SMUR, but it's a different class of E3 ligases. It's a hacked, it's not E3, forgot it. So it's a different class of E3 ligases.
CBLLB is one such ligase, ubiquitin ligase, which is increased with skeletal unloading. Okay. So, and what I'm blocking senioritis on the author, but he's shown it in both muscle and bone. What it does in bone anyway is lead to the degradation of IRS1, and apparently it's also a target in muscle, although there are other targets in muscle.
I just wanted to mention another resource, and that is there are people like Jake Lucis and Charles Farber who have done expression profiling of different mouse, like 90 different mouse strains, along the lines where they're, they know the muscle and bone, and there's expression profiles available, and those are going to be very interesting to go look at. Where again, just like Dr. Kiel showed the genes that are there, but this is expression. And I think that's the other level that you need to take. And yeah, it's a mouse, but you still, I think it would be very informative to start looking at those expression profile overlaps.
Yeah, when you start trying to assemble the tissues of interest, the sample sizes are rather small. And we're hoping, with time, people getting muscle biopsies and actual whole bone expression is going to be of a sufficient size to be able to make something out of the functional significance of the findings.